No voice.
Sorry, what?
I thought I'm not unable to hear. That's why I said no voice.
Oh, no, no, I hadn't started. A few people are waiting, so I'm waiting for them to join.
Okay, madam. Don't worry about the lobby. I will admit all.
Okay. Okay. So, good afternoon, everyone. Welcome to the last session for this week, for this course, in fact. So, today we will be following the similar pattern. We'll first go over the previous week's assignments. We will look at the topics that were discussed as part of week 8. Try to address some questions if there are any, and we'll try to resolve the questions by this session, or if required, we can get an extra session arranged also if that is necessary. And we'll try to wrap up whatever has been discussed as part of the course till now. So if you have any doubts from the previous sessions as well, you can bring them up and we'll try to solve them out today itself. And finally, we'll look at some sample problems for the week 8 assignment, and then you'll get questions for that as well. So, as part of week 7, if you remember, we had started our discussion on the endomembrane system. We had extensively discussed the role of the endoplasmic reticulum and of the Golgi, and how each of them have different roles, not just with respect to vesicular transfer or vesicular trafficking or transport of resources within the cell, but also with respect to their communication to the cytoskeleton, their roles in metabolism, etc. And we had briefly understood how they are all connected among each other, how different functions are carried out by both the ER and the Golgi, and each of them has a role to play in ensuring that whatever protein is fated to be in a particular region within the cell, whether it be the plasma membrane, whether it is within some particular compartment, all of that is trafficked and organized at the level of the ER. And we had followed that up with some introduction into how the peroxisomal proteins, not peroxisomal, sorry, the lysosomal proteins, and how vacuoles have a role to play, since they are also a part or an extension of the endomembrane system, and we had tried to wrap it up all together. So based on those discussions and what was already a part of your week 7 topics, these were the questions that were asked. And among them, the first was that the glycosylation on proteins and lipids occurs in A, lysosomes, B, mitochondria, C, cytoplasm, and D, Golgi apparatus. So while we did not get into extensive details about glycosylation, if you remember, I had talked about the fact that a large percentage of proteins, intracellular proteins, under normal circumstances require glycosylation to not just to function the proper way, but also to have the necessary structure that will allow them to function in their proper way. So the proper structural organization, folding, and of course by extension their activity within the cell is dependent upon appropriate glycosylation. And this glycosylation can be of two types. It can either be O mediated or N-linked or O-linked, which is via asparagine or via serine threonine. So this is another separate topic of discussion, but for now, you can just keep this in mind that this is the major uh, or the basic way in which glycosylation is carried out, and the compartment or the region where glycosylation occurs is actually taken care of by the endomembrane system. So while initiation of this event does occur in the endoplasmic reticulum, it is like a large sugar structure. I think there was a schematic representation of it in the previous week as well. A large sugar structure containing glucanac, which is the glucose, like derivatives of glucose, mannose, etc. They are all added at the level of the ER. And in subsequent steps, which are processing steps that are very, very essential for a protein to have the correct glycosyl substituent attached to itself, in these subsequent steps, some specific residues from that large entity are removed or are snipped off at each stage. And this is carried on in the ER and further taken care of in the Golgi. In fact, majority of it does occur in the Golgi as well. So this glycosylation is taken care of by these two endomembrane compartments. cells. Now, as part of the question, since we don't see that endoplasmic reticulum is an option at all, and Golgi has a major role to play in it as well. So the glycosylation can be attributed to the Golgi apparatus inside the cells. The second question is about the organization of the Golgi, which is in the form of dash in normal adherent mammalian cells. So we have been asked that what is the pattern of organization. So let us look at the options first. First option dedicates that it is a membranous stack of cis, medial, and trans Golgi cisternae. Second is that a number of vesicles are just scattered haphazardly in the nucleus. Third is that the double membrane structure is present, which is internally lined by cristae, or is it a tubular network that is spread throughout the cytoplasm? So if you consider a normal adherent mammalian cell, then obviously we know the correct option to this are the formation of those cis, medial, and trans Golgi. The option itself is quite self-explanatory because it does mention Golgi by itself. But yes, even if you don't know that, you will remember the structure that we had talked about, that the side which faces the nucleus is the cis side, whereas the side which faces the plasma membrane under normal circumstances would be the trans side. So a protein, while it undergoes processing, or not just protein, but any cargo, when it is undergoing the processing from ER to Golgi to its predetermined destination, it actually has to go through these subsequent steps where each of these modifications will take place through the cis, then medial, and then trans Golgi. And this is the directionality of movement, which I think we had also discussed in the previous class, that there is anterograde movement and there is retrograde movement of some cargo depending on what its ultimate fate is supposed to be. So therefore, the first option is the correct option. If you look at the other options that are present, number of vesicles scattered inside the nucleus, certainly it is not true. Third option is that the double membranous structure, which is internally lined by cristae, this is actually true for mitochondria. So if you have looked at the ultrastructure of mitochondria, it does have a double membrane structure, and it has these sort of zigzag pattern of cristae which are around. Those structures are known as cristae. They're a little bit like crypts that are present inside, and that composes the mitochondria as you if you just section it through the center. The next option is tubular network, which is spread throughout the cytoplasm. This is more true for the endoplasmic reticulum. It does have a tube-like structure, and it is quite extensive. The structure is not compact. It is quite extensive, and a large portion of it is actually in direct contact with the nucleus because of the things that we had discussed the last day, that a protein after not a protein, rather an mRNA, after it has been synthesized in the nucleus, is brought into the ER for further subsequent processes to be take place, which also in a large number of cases involves the process of translation, which is the synthesis of the polypeptide chain, which will then undergo processing. So these are all descriptions of other compartments for Golgi. The first option is obviously true. Then in option 3, we are asked that which of the following statements about the endomembrane system is true. So we are talking about the endomembrane system. Among them, the options are A, the mitochondria and the Golgi apparatus are part of the endomembrane system. No, mitochondria is not a part of it. In fact, it's not believed to be a part of it because it is not made up of similar structures or similar lipids. Its origin is something that is questioned, while in the current scenario, of course, the origin is from the parental cell to the daughter cell, but it does have an endosymbiotic origin that is often associated with it. So it is not a continuous part of the endomembrane system as we have understood it till now. Second is that the secretion of proteins is one of the major functions of the endomembrane system. This is in fact true, and this is something we have discussed as well, that secretory proteins need to undergo this processing, and they need to go through the processing through the plasma membrane, and only then can they be sent outside. So it is a part of the endomembrane system and the proper organization and functioning of this system as well. Then in option C, we are asked that the Golgi apparatus can target vesicles to all sites in a cell, including the endoplasmic reticulum. It in fact can. it might seem so that, when a protein has undergone processing already in the ER, if you look at it as a flow chart, you will think of the nucleus, then the ER, then the Golgi, and then the plasma membrane. This is the route that is followed by a protein that is intended to be secreted outside. For a lysosomal protein or for a mitochondrial protein, or not a mitochondrial, but any other compartment where it is supposed to be directed to, it will have to undergo through this, or it will have to go through this flow chart where the ER comes first and then comes the Golgi. But the fact of the matter is that ER itself also has a large number of proteins. Like, for example, one that we have already encountered was the translocase. That translocase was localized in the membrane of the ER, and it was necessary for that translocon to be present there to allow the polypeptide chain that is being synthesized to allow its entry into the ER. Translocase was required. Not just that, within the ER, there are a large number of enzymes, particularly, which are known as chaperones, which are assistants, like they're not assistants, but these are they are helpers. You can think of them as helpers, which allow the proper folding of proteins when a protein has undergone misfolding. So it's a complicated concept, but for now, this in itself would be enough to understand. I think that they just provide a surface on which a incorrectly folded protein can sit and can correctly fold to its functional conformation. Now this now this is part of these proteins that need to be redirected back to the ER. There are many other proteins as well, but for this redirection, the Golgi has another set of transport that we just talked about a little while ago, which is the retrograde transport. That is, while the normal transport is occurring from cis-medial to trans-Golgi during the process of this pinching off and fusion with the next vesicle, there is a separate system operational as well, which also includes a large number of other proteins, where it has to go through the trans, to the medial, and then to the cis Golgi to be redirected via certain other helper proteins to be redirected into the ER. So there is a separate system which is in place for facilitating this as well. You can read it up. This entire process of vesicular trafficking is a complicated topic by itself, so we have not gone into it, but there is you can find it in books as well, and of course you can refer to research papers if you're interested. So yeah, so this is what we were talking about. So both options B and C are correct, and therefore for question 3, option D would be correct. Then coming to question number 4, which of the following statements about the endoplasmic reticulum is false? First would be that post-translational modifications of proteins begins in the ER. This is in fact true because, like I said, glycosylation, it is beginning in the ER. Glycosylation is a post-translational modification. Please keep in mind that whenever you're answering questions like this, it's very easy to get the terminologies incorrect. You can have post-transcriptional modifications, which is occurring to the mRNA, and that includes splicing, polyadenylation, and some other modifications as well. However, when you talk about post-translationals, that is when the polypeptide has already been synthesized and that is undergoing changes. So one of those changes is the addition of the glycosyl group, and many other glycosylations also occur. One common example, if you need to think of glycosylation, is for antibodies. So antibodies, if you are familiar with, they are already they are polypeptide chains basically, or proteins basically, and they need to carry the glycosylation because it is essential for them to be soluble within the physiological system. So that glycosyl group is crucial for their solubility, and therefore it just shows that how the attachment of a simple group can really have so much impact on how functional a protein may or may not be within a system. So this is one example of it. So post-translational modifications of protein, it does begin in the ER. So option A is correct. Second is that rough endoplasmic reticulum is associated with ribosomes, which is also true. It is in close attachment with the nucleus, and it does carry ribosomes, which gives it that rough exterior, which is how it is visualized and how it probably has been named as well. So option B is correct. Option C is that ER is part of the endomembrane system itself, and certainly this is correct. So yes, the option D is the default. Yeah. Option D is the default correct answer for this case. So none of the above statements are true, which would be the correct option here. Then in question 5, we were asked about the smooth endoplasmic reticulum. So the SCR is which of the following? Is it just the endoplasmic reticulum that lacks ribosomes? Is it the site for glycosylation of proteins and lipids? No. Is it abundant in cells synthesizing proteins? Not necessarily. I think we had discussed this in the last session as well, that for a cell that is very actively undergoing protein synthesis or that is very actively carrying out protein synthesis, what needs to be very obviously when you talk about synthesis of proteins, the molecular machinery, which needs to be available in abundance within the cellular environment has to be the ribosome. So when the ribosome is enriched, you expect that the enriched subcellular compartment within the cell that would be present in abundance is the RER, which is the ribosome associated endoplasmic reticulum. So that would be option 3 would be true for rough endoplasmic reticulum. And fourth is the site for microtubule nucleation. We had seen that Golgi is in fact capable of doing that. And in fact, Golgi anchored microtubules are also a subclass that we had looked at. Therefore, from these 4 options, the correct alternative to choose seems to be the fact that SCR is just the endoplasmic reticulum that lacks ribosomes. So yeah, that was question 5. In question 6, we were asked that which of the following is not involved in the regulation of Golgi organization. So we have chaperones, microtubules, ARF, and CTBP1 or bars. So I think we had looked at the other 3. I'm not sure if we looked at CTBP1 or bars, but from these options, chaperones are certainly not involved in organization of Golgi. In fact, I think I just introduced chaperones to people. So chaperones are not more, it is actually their responsibility or their function to carry out the correct folding or rectify the misfolding of proteins that have somehow undergone that inaccurate manner of folding in some cases. So that would be the correct option for question 6. In question 7, we were asked that lysosomes are called as suicide bags of the cell because A, they can digest carbohydrates, they carry hydrolytic enzymes, which when released into the cytoplasm are capable of killing a cell, or whether they can fuse with endosomes so they can recycle the waste that is generated in cells. So this was also discussed in the previous class. So if you remember, suicide bags or lysosomes within the cell are named so because they are very, very rich in these enzymes, and these hydrolytic enzymes, lipases, hydrolases, etc. All these ases are enzymes which are capable of breaking something down. So that breakdown under certain circumstances is, while under normal circumstances, it is expected to help in the breakdown of whatever resources are being procured by the cell to provide energy to it and to remove any sort of undesirable substances as well. However, under certain circumstances, which under certain circumstances is the only option that the cell has when it is under extreme starvation or some other hindrance has come about, then the cell decides to break its own compartments or break its own resources in order to survive long term. So that is autophagy, which we had introduced, and therefore hydrolytic enzymes can often end up killing the cell when it is required, when they're released into the cytoplasm. And that is why lysosomes are called as suicidal bags, because they are killing what they are themselves a part of. So that is option B. As part of question 8, we were asked that mucolipidosis 2 is a lysosomal storage disorder, which it's a lysosomal storage disorder which is caused by the loss in mannose-6-phosphate signal that targets enzymes to lysosome. Yeah. Okay, this is just an example which has been given here. So it is an lysosomal storage disorder where mannose-6-phosphate is lost, and this mannose-6-phosphate signal under normal circumstances is responsible for lysosomal targeting of enzymes that have been synthesized. And when this is not present, it is often leading to this particular disorder known as mucolipidosis 2. The question is that the dysfunction of which cellular compartment or which cellular component could be the cause of this disorder. So, what could possibly be the reason for this? Which is where you see that there's a loss in mannose 6-phosphate signal. So, this could be because of some error within the Golgi apparatus. Because, like I just mentioned, that there is some sort of processing or snipping that is occurring within the Golgi as well, and just give me a second. Okay. Yeah, so sorry, I got interrupted. Yeah, so we were talking about the lysosomal storage disorder, and the question was about the dysfunction in which of these components could be the cause of this disorder. As options, we have peroxisome, mitochondria, Golgi apparatus, and vacuoles. Now, like we had just discussed, that glycosylation and its processing is taken care of by the ER and Golgi, particularly the processing of the attached glycosyl group that occurs within the Golgi apparatus. And therefore, we can expect that while under normal circumstances, you would expect a protein that is destined to go into the lysosome to carry the mannose 6-phosphate signal. If there is some sort of dysfunction within the lysosomal and within the Golgi enzyme that snips off these glucose 6-phosphate, mannose 6-phosphate residues, it can lead to a condition where where the signal peptide, which is where the signal for the lysosomal uh, destination, which is mannose 6-phosphate, is also getting cleaved off. And what will happen as a consequence, you can expect, is that the protein will no longer carry the signal to be destined for any of the compartments and would by default accumulate within the cytoplasm. And as you can expect, that would have very far reaching, very detrimental consequences, because if that enzyme somehow is somehow figures out a way or somehow is able to function under the physiological pH, it can lead to very drastic consequences, and that would certainly not be something that could be salvaged by the cell at that point. So it would be a very severe disorder. And if you're interested, you can certainly go and look up a little bit more on the disease. You'll find a lot of different disorders, not just lysosomal storage disorders, but also peroxisomal diseases, which are not that common. They're very uncommon, but um, they are prevalent, and they are very detrimental to the individual that suffers from them. And um, not much has really been discovered as a solution for such problems. But yes, there is ongoing research about them, and if it interests you, you can certainly go and look it up. Um, Coming to the last two questions of the previous week's assignment. As part of question 9, we were asked that cell sap is A, the cytoplasm of plant cells, B, the fluid in plant vacuoles, C, the fluid in the nucleus of plant cells, or D, the lumen of the plant ER. So cell sap, as you can understand, is part of or is the component fluid which is present within the vacuoles of plants, and therefore option B is correct. And finally, as part of the last question, was that which of the following is the correct route followed by a endocytose product? So I think I just mentioned this a while back as well, that if you could just summarize the entire process of vesicular trafficking into sort of a flow chart, then what it would look like is that it goes through okay, so keep in mind that this is not just starting from nowhere, but it is the path that is being taken up by an endocytosed product. Just give me a second. Option is correctly marked. Please.
Madam, two doubts. Question number 5 and question number 10.
Sorry, I couldn't hear you.
Uh, There are two doubts regarding question number 5 and question number 10.
Okay, come to question 5 first. Yeah, go ahead.
Smooth endoplasmic reticulum. The option correct is endoplasmic reticulum that lacks the rhizosome. Actually, what is in
Mm-hmm.
my mind, endoplasmic reticulum, there are two types. One which
Huh?
smooth with ribosomes and without uh, ribosome is a with ribosome is a rough endoplasmic reticulum, and in between that is
Right.
they call it as the transitional ribosome. Okay, madam. Why I confuse is because endoplasmic reticulum is a site for glycosylation and lipid synthesis, and I don't know whether smooth is important for protein synthesis or function for the protein.
No, so
Actually, that occur in
smooth
endoplasm
endoplasmic
is
reticulum.
true, but whether it is present on only rough endoplasmic reticulum, that I don't know, because I think rough
Okay,
ribosome and
so
protein, these are different things. It's macromolecules
they
now
are, of
we
course, they are.
divide into four parts: protein, lipids, carbohydrate, and nucleic acids. Ribosome, I think, is a part of nucleic acid. Means second option is also correct site for
No,
glycosylation of lipid.
ribosome. No, no, no. You are not correct. Hold on. Let me explain it. First of all, the first thing that you said that ribosome is not nucleic acid. Ribosome is a huge macromolecular machinery. You can look at the structure, which is made up of 99%, if I'm not, I think I might be wrong about the statistic, it might be 95% or whatever, but it is a protein machinery. There are a huge number of proteins. There is a large subunit, there is a small subunit. We have discussed all of this very early on in the second or third week. We have discussed it. The large subunit is made up of proteins which are labeled as L. There are different sedimentation coefficients of each protein. A large number of proteins form an entity which is the large subunit. You have a smaller subunit which is right below it that also carries a certain number of proteins, relatively less number of proteins, but that is what the assembly is made up of. That is a ribosomal machinery. What it has in apart from these are some rRNAs, which we have talked about. And those are for small subunit, you have a 16S rRNA. For the large subunit, you have 23S, 5S. You have all of these rRNAs. So rRNA is a very small portion of the ribosome. The function of the ribosome has to be carried out by the rRNA, but it cannot do so by itself. If you just put these rRNAs into any system, they will not be able to recognize an mRNA and synthesize a protein out of it. No, it requires the enzymatic activity while it lies with the rRNA. It requires the protein to accompany it to form the composite system, which is the ribosome. Only then it becomes functional. So I know that was not your question, but I wanted to clarify it that you cannot classify ribosome as any one of these XYZ things. No, it is an assembly. It is known as a ribonucleoprotein complex, RNP. There is ribonucleoprotein, there is ribonucleic acid, and there is protein, which are together forming a complex. That is how it works. You can classify other enzymes, like you can call DNA polymerase or RNA polymerase as just proteins, and there is no debate about that. They are proteins. They are proteins that have enzymatic activity. In this case, both are present. Okay, that part is now separated. Coming to your question, what you're saying about transitional endoplasmic reticulum, this concept was not a part which was introduced in this course. Okay? Based on what was taught in this course, they have just talked about rough and smooth endoplasmic reticulum here. Very directly classified them into these two categories. Now, based on these two categories, the first distinction between them is in terms of their appearance. That appearance is coming because of the presence or the absence of ribosomes. When ribosomes are present, you're able to see they are forming a rough surface and they are capable of protein synthesis. In comparison, for the smooth one, you do not see the presence of ribosomes in that case, and therefore they are appearing smooth to you. Now coming to how they are functioning. When it is directly, when the part of the endoplasmic reticulum, because it's a tubular network. It's not one compartment, right? I think I mentioned this also in the last class. Because it's a tubular network, a part of it is in direct contact with the nucleus, which is where transcription is occurring. So from transcription, the immediate next step that under normal circumstances is required is translation to be carried out. For that, you need ribosomes to be available. That availability is brought about by the endoplasmic reticulum that is carrying ribosomes, which is why RER is in direct contact. Most cases it is in direct contact with the nucleus. So that is the function of the rough endoplasmic reticulum, to provide the ribosome, which will allow that to take place. But that is not the only place where ribosomes are present. They are present in the cytosol as well. A large part of the mRNA which is being synthesized is actually bound by the cytoplasmic ribosome, and that cytoplasmic ribosome, it comes on the ER, sits on the translocon, and allows that mRNA to be translated, and for the synthesized polypeptide to enter into the ER. That is how that function is taken care of. When we talk about the smooth endoplasmic reticulum, its job is not to do this. Its job is not to carry out protein synthesis. So that option is automatically getting eliminated. If I come to the option, let us ignore the first option for now. The second option is that site for glycosylation of proteins and lipids. No, under it is not where protein synthesis is occurring, or protein glycosylation is occurring. Most cases when you have a um, when you have a ribosome on the surface, that ribosome is bringing the mRNA, and that mRNA is getting translated into the protein, right? Only then that protein is able to undergo the process of glycosylation, and or lipid glycosylation, whatever might be the case. So that function is also taken care of by the RER. Smooth endoplasmic reticulum has roles in some cases in lipid biosynthesis and in lipid catabolism as well. I think if I'm not wrong, omega or alpha, one of these two types of lipid breakdown or lipid catabolic processes, they take place in the smooth endoplasmic reticulum. I think it is alpha oxidation which takes place. Like there are different types or different mechanisms by which lipid is broken down inside the cell. One of those mechanisms is taken up by the smooth endoplasmic reticulum. Please look it up. I also need to brush on it again. I think it is alpha or omega, one of these two, that is taken care of by the SER. That is its job. Smooth endoplasmic reticulum is not responsible for carrying out protein synthesis, protein glycosylation, lipid glycosylation. No. So these three options are automatically not correct, which is why you are coming back to the superficial classification of the two, that smooth endoplasmic reticulum is becoming the ER, which is lacking the ribosome. I think that is how you can interpret this question. Please let me know if I created more confusion or you would like some more clarity.
Thanks, madam. Now, question number 10.
I have a doubt for question 10 myself, but yes, please go ahead.
What is endosome, and does endosome and Golgi vesicles, they are same or different?
No, completely different.
Vesicle is a part of
Golgi
endosome,
is.
or it's not part of endosome? That's what my question is.
Can you repeat the last part? I couldn't hear you.
According to me, endosomes are rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, and vesicles. So that's what I'm asking. Ki Golgi and vesicles, these are part of endosome or not a part of endosome?
No, okay. So I think the way you're looking at it is not exactly the right way. So what you need to understand is, for any cell, these 3-4 organelles, they are always present. So ER, Golgi, and plasma. You can think of the plasma membrane not as an organelle, it's a boundary of the cell. But these are cellular components. They have definite functions inside the cell. Okay? They are organelles. They are present irrespective of what type of cell it is, because they have some definite functions that is necessary for the cell to survive. The endosome by itself does not have a function. It is not carrying out protein synthesis or in anything of that sort. It does not have any survival essential function. What is happening is, you need, let's think of it this way. Think of a differently abled person when they are entering into a unfamiliar environment, right? Not the most appropriate example, but just take it as an example for now. When they're entering an unfamiliar environment, in this case, that is some sort of resource for the cell. It is some sort of a resource which has to be brought inside the cell. For its entry, because that person is not able to enter by themselves, you need a carrier. You need some sort of a package or some sort of a protective environment for such an individual. So in this case, that package is an endosome. That endosome is simply a membranous sort of structure. So when you have the plasma membrane of the cell, or the cell membrane, whatever it is, when it is encountering a food product, so imagine this is the cell membrane under normal circumstances, and there is some food product which is coming here. So what the cell membrane will do is will try to engulf whatever food product is there. When it engulfs, a part of it pinches off. So you had a continuous system. From there, a part of it got snipped off or a part of it got pinched off. And when it is pinching off, it is taking whatever food resource there was into that package or into that compartment. So analogous to the example that I was giving, that if it is a person who is not capable of traveling by themselves in the cellular environment, because the cell's interior is not something that the food product is supposed to be in. The cell's interior is very strongly protected. It has a definite pH, it has ions, it has a very complicated architecture inside. So nothing can just enter as and when it wills to do so. So it has to be protected, it has to be brought inside an endosome. That endosome is part or is pinching off from the lipid bilayer, which is on the surface. Once it is brought inside, it is going inside doing whatever its job is. If it is to be broken down, if it has to go to the lysosome, then it has to go there, it has to be broken down. Whatever has been broken down and whatever has been generated, say it is some sort of an amino acid, or it is some other resource for the cells. If it was a proteinaceous source, you got amino acids. That amino acid will then have to be delivered to different parts of the cell where amino acids are required. Where is it? It is required for protein synthesis, right? During the process of protein synthesis, when tRNAs have to be charged, that tRNA will have to be attached to its designated amino acid. So from the digested product, from the lysosome that has digested whatever food resource there was, these amino acids are made available. So that is how the circulation within the cell occurs. That food resource is brought in, it has to be broken down, and it has to be utilized by the cell. So that is the job of an endosome, to provide that carrier, to provide that mediator through which it can be transferred or it can be moved around within the cell. It is not a part per se of any of these organelles. When it is pinching off from the ER, this I just gave the example of a lipid. If it is pinching off from some other membrane of the cell, or it is, say, think of it in this way, that when autophagy is occurring, right? Under those circumstances also, you are seeing there is a fusion of vesicles which is occurring. There is a phagosome, there is a lysosome, there is an autophagolysosome. Lots of structures are happening. These are just simple lipid environments that are fusing with each other and making two compartments into one. So endosome is also a lipid compartment. Think of it in this way. Wherever it pinches off from, that is what it is serving as. It is a vesicle, or it is a compartment, not a part of any of these organelles, or not specific to any of these organelles. That is my perception of it. Please let me know if it is clear or not.
Madam, just I want to know whether lysosome is a part of endosome, or there can be endosome. If you, what I got from you is endosome is for a temporary structure, which can be unit, which can be multiple, which can be early, which can be late endosome. That's okay,
Yes.
but what about the lysosome and Golgi? Because endosome is a part of vesicle. Because what question is
Hmm.
there? Endocytosized. Endocytosized product.
Endocytosed. Endocytosed. Yes,
Endocytosed
endocytosed
product.
product is anything which
What
is
is
taken up by
what
the cell from the outside to the inside.
is it? So either it will be breakup from vesicle, or it will be breakup from endoplasmic reticulum, or it will be breakup from Golgi apparatus. So that once it is
No, it will
endocytosed,
break from the plasma membrane. That is what I explained, right? When you
or
have to understand what is the
it can break from
process.
plasma membrane, means all four things, but its root will be then plasma membrane, endosome, and then again plasma membrane. Why Golgi apparatus, the vesicles they are involved? That's what confuses.
Okay, that is the okay, okay, got it. So see, one thing you need to keep in mind is okay, it has formed from the plasma membrane, it has formed the endosome. Say suppose that food product is something that is going directly to it could also be a way that it could have gone to the lysosomes. It could have gone to any other compartment, right? From my understanding, how you can answer a question like this, because to me also it looks a little bit ambiguous. I think there could be better combinations that could have been used for this question. But for now, what you can understand is because the question is asking for the process of endocytosis, the first step that has to occur is that the fusion or the process has to start from the plasma membrane and it has to go from there. If you look at any of the other options, none of them are starting from the plasma membrane. So for the product to be endocytosed, you start with plasma membrane. It is brought into the endosome, and in my opinion, there should have been a step of lysosome here. It should have been broken down here. And then from lysosome, there should have been vesicle and then the plasma membrane. That would be the ideal option, in my opinion, as well. But what you can, when you are seeing that none of the options are fitting into what you know to be the correct one, you can go for the best fit. And among the best fit, it has to start from the plasma. I am sure you agree on that as well, that it has to start from the plasma membrane. If in case the process was exocytosis, where something from the cell has to be pushed out of the cell or has to be secreted out, under those circumstances, what your priority should be is that you have to go for the option which ends with the plasma membrane. Last fusion should be at the plasma membrane. I hope that makes the distinction a little bit clear. That if something has to be taken up, you start from plasma membrane, you go to vesicle, you go to whatever organelles are there inside the cell, and then you again recycle it back to the plasma membrane. I am in agreement that there could have been a better option, but among these, the best fit is option C.
For solving MCQ, it's okay, but what about the endocytosed product? Actually, what happens to it? Means from gaining the knowledge and
Depends
solving
on
the
what is.
MCQ, these are two different things. Means that is a
I
lesser
agree. I agree. So
but I agree with you.
yes.
But created confusion really endosome, Golgi, and vesicle. These there should not be three options. There should have been only one option: endosome, plasma membrane, endosome. That is my actual thinking was.
No, but if that is what is happening, okay, let us assume that was an option. But then what is happening to the product? If you just say, suppose the cell is procuring a food particle from the outside. If it is just going from the plasma membrane to an endosome and then back to the plasma membrane, then what happened to it? What happened to the food product? It got wasted, right? Whatever food product was there, it has to be taken up by the cell. If you just circulate it inside and out, then that is becoming a pointless expense for the cell, because each of these processes is costing energy for this simple food product to be brought in. There are proteins, there is, I think there was some a schematic for that as well, that there are so many proteins that are investing energy to bring it inside. So that food product has to undergo digestion, has to be broken down, it has to be used by the cell. And after that, whatever is not required, the cell can flush it out. It can get rid of it. So there has to be some sort of involvement of the cellular compartments in it. I think there should have been a lysosome in this case. The lysosome should have been the organelle where it goes to for breakdown. It could be that there is some function of the Golgi apparatus as well in endocytosis that I'm not able to recall at the moment. Maybe I'll go back to it again. But just having plasma membrane, endosome, and plasma membrane will be a wasteful investment for this cell. There should be some breakdown there.
Okay, madam. Thanks.
Okay, so yeah, I think this covers the assignment questions. So we'll just go over, I think we are already 1 hour past the class, so we'll go over this week's topics a little bit quickly. Okay, so as part of week 8 as well, endomembrane system was only continued, but among them, the focus was shifted a little bit more towards the nucleus. It has been explained very well in the lectures, so I will brush over the topics and I'll get to the sample questions as soon as possible. So coming back to this already familiar diagram, if you remember, we saw that there is the nucleus. In attachment, there is the rough endoplasmic reticulum, the smooth one, the Golgi, and the external plasma membrane. And interspersed, you see lysosomes and secretory vesicles, which are present at different locations, or not random, but at certain positions inside the cellular environment or the cytosolic environment. Next, the focus here, it moves towards it zooms in more towards the nucleus, the nuclear membrane, and the nuclear environment more so. So what you see here is that there is a sort of sheet-like structure that you see, if I think it is visible. So this green structure that you see everywhere, these are little sort of like a mat or a mycelial mat-like structure of the endoplasmic reticulum, which is spread extensively throughout the cell. But apart from that, what you see is that within the nucleus also there is some sort of architecture which is present. There is a nuclear envelope and there is an architecture that is present at the nuclear envelope as well, which performs very, very important functions. We'll get into some detail about that now. Here you see that this was an image of the intracellular organelle and cytoskeletal interactions, which was shown at nanoscale resolution on milliscale times, millisecond time scales. So here you see the cytoskeletal interactions which are present, just immunofluorescence or probably a I'm not sure, but I think it's a confocal image only. So it's a confocal image of the cytoskeletal network that is present inside the cell. What is the function that we are trying to look at here is the nucleus, right? So what is the nucleus? Nucleus, as you understand, is an organelle found in most eukaryotic cells, exception being red blood cells. So this is often a question which is very frequently asked. They are often also known as ghost cells because they look like there is nothing inside. They look like they're empty, which is of course not the case, but they are enucleated cells or ghost cells within the any eukaryotic individual. In animal cells, it is both the largest and the stiffest organelle and is very easily identifiable by light microscopy. Like in some cases, if your cells are big enough, for example, if you talk about cancer cells, among them HeLa cells, which is a type of cervical cancer cell line, one of the most aggressive cell lines that is very frequently used in most cancer labs, you can very easily, without any staining, without doing anything at all, you can see the nuclei of these cells just because they are so large. You can see them very easily. Even for hex cells, they are quite easily visible. Apart from that, when you go through staining procedures, of course, the nucleus is the most easily distinguishable part of it. And apart from that, like it has been mentioned, it is the largest and the stiffest organelle. The average mammalian nucleus has a diameter of approximately 6 microns, and it occupies about 10% of the total cell volume, which is a significant percentage for just one organelle to occupy. But then, of course, you're already familiar with how crucial its function and how crucial its spatial occupancy without the cell, within the cell, it is required, and therefore it takes up such a large part of the cellular microenvironment. So among the so we have looked at multiple cross sections of the cell. This one goes into a slightly detailed information about the same. So what you see here is that there are different subcompartments that have been labeled. We have the nucleus, the nuclear lamina, nucleoplasm. The nuclear lamina is, if you remember our discussion on the cytoskeletal network about intermediate filaments, then laminins are part of intermediate filaments and nuclear lamina is indeed made up of them, along with other components as well. Then comes the nucleoplasm, the euchromatin, heterochromatin. It is not very distinguishable here, but they're just trying to say these are the different compartments that you see inside the nucleus. Till now, we were talking about everything that is present in the cytoplasm. Now we are zooming in a little bit specifically towards the nucleus. And apart from that, we see there is the nucleolus, the nuclear envelope, the ribosomes, nuclear pore. This is, of course, outside of the nucleus. You have the ribosomes, the nuclear pore, and the rough endoplasmic reticulum, which are all present. Here you see a schematic representation of what the nucleus basically looks like. And this is a cross section of the same. So what you're seeing here is that there are the rough endoplasmic reticulum that is in contact with the nucleus, like we have already discussed. Within the nucleus, we are seeing that there is a lot of, it's very dense. It's not it does not have space lying around. It's not even a viscous sort of environment. It's a very densely populated environment. Why densely populated? Because it is so hugely enriched in nuclear proteins and the chromatin as well. So, like we talked about the protein vesicular trafficking, that there is retrograde movement towards the ER. There is, of course, also very defined movement that has to occur towards the nucleus, because the nucleus has so many proteins. If not anything else, think about histones. Histones are taking up such a large part of arranging the genomic material or the genetic material inside the cell, and therefore their correct direction back into the nucleus is also extremely important for normal function, for normal organization of the DNA inside a cell itself, and of course subsequently during the process of cell division and process of cell division as well. So apart from that, you see that there is euchromatin and heterochromatin. So if you're not already familiar with it, the way in which the wrapping or the packaging of the genetic material is done allows its classification into these two structures. What are these? That is euchromatin. Euchromatin has the less compact structure. Often it is just seen as the 11 nanometer fiber. So what is this 11 nanometer fiber? If you remember from our epigenetics discussion, very briefly we had that discussion. You remember how DNA, it gets arranged into thicker and thicker sections because of the way in which it is packaged around different structures. So the 11 nanometer fiber is often the loosely bound or the relatively free structure, which is euchromatin, and it has the appearance of beads on a string, where the beads in this case represent nucleosome, and of course the string represents DNA. Secondly, heterochromatin is more compact and is often reported as being composed of a nucleosomal array, which is condensed into a 30 nanometer fiber. So like I just said, that the more you move along the packaging of genetic material inside the cell, the more condensed or the more packaged or the more compact it gets, and it builds up from 11 nanometer fiber to a 30 nanometer fiber by just winding around the heterochromatin. I think I had shown you the demonstration as well of that mouse and the string which is wrapped around it. So that is the heterochromatin. The heterochromatinized region of the genome or of any chromosome is transcriptionally inaccessible because it is wound so tightly, transcription machinery can't sit on top of it. Versus when you talk about euchromatin, it is the less it is the more accessible or the less tightly wound area, and there the transcription machinery can sit very easily and can express those regions of the genome with much ease without needing too much of readjustment of the histone attached with it. So yeah, those were the two types. And apart from that, you see the nuclear pore, you see nucleoli, and you see the nuclear membrane also as part of the ultrastructure. Now here we see another representation of pretty much the same thing, but at a closer level. So at the nuclear periphery, you can basically see three different layers. You can see the outer nuclear membrane, the inner nuclear membrane, and the lumen between the two. You can see that there is a complex of proteins, which is known as the link complex, that is linking it or is linking the nucleus to the cytoskeleton, to the cytoplasmic skeleton. And this link is carried out by the complex known as the link complex. Now, how is this link being executed? Right beneath the nuclear membrane, there is an extensive network of lamina or lamins, which is forming the nuclear lamina. So if you consider a cell, for the cell membrane, right beneath the cell membrane, we had seen that there is an actin meshwork which is present. We talked about actin sheets, actin bundles, actin networks, etc. All of these to be present right beneath the cell membrane. If you just take that analogy and look here, you will see that beneath the nuclear membrane, you have this meshwork of um, nuclear lamina. And this nuclear lamina is the nuclear cytoskeletal architecture that is being linked to the cellular or the cytoplasmic skeletal architecture via the link complex. That is its job. Right beside it, we can see the nuclear pore complex. So there are pores or regions of transport within the nuclear membrane where they allow whatever proteins are inside or whatever is synthesized inside to go outside, and whatever is outside to come inside based on some very specific import-export mechanisms that are there. And this is mediated via interaction with the nuclear pore complex or the NPC. And apart from that, we are seeing that the inner nuclear membrane proteins are very yeah, so the nuclear pore complex is responsible for interacting with whatever cargo is being moved inside and outside the cell, and whatever proteins are responsible for mediating it as well. And right next to the nuclear lamina, we can also see the endoplasmic reticulum. And that, of course, like we have discussed previously, is the RER, as you can see in the feeds on string structure or the simply the black and white image that you can see on the right. So it is representative of what is happening inside the cell as well. So here you can see that there is again, this is a part of the discussion we've already had, the rough ER and the RER, how it is present. And this is the detailed architecture of how the nuclear pore complex is formed and how the link actually operates in this context as well. So you don't need to remember everything, but the basic idea would be helpful. There is a lot of details that can be gotten about regarding these topics. But for now, what we can see that there's a nuclear basket. That basket is towards the nuclear side. So whenever we talked about the cell membrane, we always talk about the cell cytoplasm and we talk about the cell exterior. With respect to the nuclear membrane, there is the nuclear side and the cytoplasmic side. You need to make these orientations very clear in your mind. And in that context, you can see that the part of the nuclear pore complex, which is towards the nuclear side or the inner side of the nucleus, is the nuclear basket. You have the inner ring, you have a transmembrane ring, and then you have an outer ring, which is linked to cytoplasmic filaments. So that is how the pore complex is formed. And at the center, you have a sort of schematically represented transporter-like structure through which whatever needs to be passaged will undergo the passaging or movement. On the right hand side, what you can see is the nuclear skeleton and cytoskeleton, which is being linked by the link complex. So on the nuclear side, you can see the nuclear lamina, which is being connected by certain proteins like SUN1 and SUN2, Nesprin, plectin, etc., to the actin filaments or to intermediate filaments within the cell. So again, this is a little bit of extra detail, but if you want, you can remember it that the proteins that link the nucleus to the cellular actin are nesprins, and those that link it to intermediate filaments are plectins. So too much detail, but yes, for your understanding, you can keep it on mind. So here what you see are the different types of proteins that are involved. So you can see that there are cytoplasmic transmembrane proteins. NUPs are basically nuclear proteins. And the different types of proteins in vertebrates as well as in yeast have been mentioned. There are different types of NUPs, which are inner ring, outer ring, linker NUPs, central NUPs, transmembrane ring NUPs, and the cytoplasmic ring NUPs, etc. So all of these, this entire organization is made up of proteins, different types of proteins with different modifications, each having some specific job at the site of the nuclear pore. So here what you see are the sort of what do I say? The realistic image of what is happening inside the cell, a space filling model, but more realistic than the representation that you were seeing before. And this is how the nuclear pore complex actually looks. So there are 28 flexible connectors that tie the inner ring of the complex together and that are likely to aid the rapid assembly and disassembly of the nuclear pore complex at the time of cell division. This is necessary. Why? Because you're looking at such a huge assembly of proteins, and when a cell divides, it has to make sure that it is completely dissociating those protein complexes, and when it is again forming two daughter cells, it is able to undo the same. So this actually brought about the question that are all NPCs or nuclear pore complexes in a single nucleus, are they essentially the same? The answer is a no, rather a definitive no. And what you can see here are that there are three very distinct variants that are present, which are capable of coexisting within a single yeast cell. And each of them has different sort of functions. They are responsible for transporting different types of proteins or different types of RNA. And based on whatever their function is, there is possibly some sort of specialization that exists inside that exists at the level of the nuclear pore complex. So the lamins that are distributed throughout the nucleoplasm, the lamins that are situated or distributed throughout the nucleoplasm have been shown here. Humans majorly have two types of lamins. We have A types of lamins consisting of lamin A and lamin C. Primarily, you see them in cells that have already differentiated. And the ones that you see, the the B type lamins, which are lamins B1 and B2, are found to be present in all nucleated cells. So this is another representation of the same thing. You can see there are so many other proteins that are present that are associating with the chromatin, with the nuclear pore, with the lamins, etc. So you can see there is RB, there are PCNA, RFC, Pol complexes which are involved in replication with the chromatin. And you can see there is some sort of unwinding here at the level of the chromatin as well. So lots of the nucleus, that the way that we usually draw the nucleus is not how it actually looks. There is so much control that is occurring, so many proteins that are working at each stage to make it all feasible. So that is quite a huge idea to comprehend and actually understand. But yeah, that is at the level of lamins. And in fact, here, as you can see that different mutations in lamin A are associated with remarkable array of diseases. So you can actually go and look at some cytoskeletal disorders if it is something that interests you. Intermediate filament disorders particularly would be useful. In fact, some proteins, some intermediate filament proteins are named, or their nomenclature has come from the sort of disease that they cause. Like there is NF1, NF2, those are neurofibromatosis 1 and 2. So these are some of the diseases that they cause. You can look at lamin A associated disorders. You can check whether there are disorders associated with actin and with microtubules as well. Look it up and see what sort of results you get and whether there are treatments for these type of diseases at all or not. Um, One example that has been shown here is of the premature aging disease, which is triggered by the dysfunctional intermediate filament function. So it leads to progeria or Hutchinson-Gilford progeria syndrome, where, as you can see, because that individual does not have the appropriate architecture to support the mechanical strength of cells that is required in an individual who is growing, they often show symptoms. They and the disease is usually characterized by what is visually seen, which is premature aging. And this was also the concept that was shown in one of the movies a few years back, which was Spa. So this actually showed that these intermediate filament proteins, they often have a role to play in the mechanical strength or mechanical stiffness of cells inside the cells inside a tissue or of a tissue at large. So the mechanobiology of cells is a whole other field that is quite interesting, that is being studied to a large extent, and that actually gives you perspective about some disease or some disorders as well. So you can see that there is a comparison here that is shown for between mucus, brain, liver, lung, muscles, agarose, bone, glass, etc. Their elastic moduli are being shown, and you can see how they actually differ over time and how the mechanics of the tissue is actually having a role to play in the development of the brain, the sort of diseases it can cause, and what how homeostasis under normal circumstances is actually maintained. So I think this shows pretty much the same thing. It's probably not something that they went into. I will move past this slide. I'll come here. So here the idea that was explained here was that the nucleus actually probably has some sort of role to play in the mechanotransduction. It basically means that the nucleus probably has some capacity of sensing mechanical stimuli surrounding the cell. So what do we mean by that? There is an example which was given here, which was with respect to blood pulsing through our blood vessels, basically, and its movement is not continuous, not like a flow, not in that sense, but instead it is sort of a pulsating movement, which is why you can feel it in terms of a pulse. And because there is this sort of irregularity in the movement, or this sort of interruption in its movement, this can actually be sensed by the nuclei of those cells which are lining the blood vessels. And that sensation has a role to play in ensuring a lot of different functions. So depending on what is the extent of lamins that are present within the cell, there can be a differential sensitivity to the movement of these. And therefore, the nucleus actually by large has a role to play in understanding and sensing and understanding mechanotransduction which is occurring within the system. Uh, then actin in and around the nucleus when it is studied. So this we talked about the intermediate filaments, particularly the lamins in context of the nucleus. Coming to the actin around the nucleus, what you see is that very close to the nuclear envelope, you're also seeing actin, more so on the cytoplasmic side than on the nucleus side. And it is believed to have some sort of roles in gene expression, in the nuclei shape and in stiffness, in cell division, and also in movement of the nucleus within the cell, or nuclear movement basically within the cell. So among them, different sort of phenomena are studied, such as nuclear migration inside the neurons, which is quite heavily dependent upon the regulation carried out by actin. Then we have cell polarization to give the cell a front, that if it is moving towards one direction, then the actin reorganization or reassembly occurs in a manner which gives the cell that strength to move in that direction. Then comes nuclear deformation through constriction. So there can be, under certain circumstances, some constriction of the nucleus can also occur, and that can lead to disruption or some deformation in the structure of the nucleus. Then coming to option 4, the cell response to some sort of mechanical stress. So you have certain proteins which are capable. So one of the chemical sensors that is um, often sensed or often considered as a key signaling mechanism is calcium. So the ER actually is capable of sensing the calcium status of the cell and accordingly can take certain decisions so as to counter whatever mechanical stress the cell is encountering at that point of time. Then chromosomes can often gather at the mitosis. At the time of mitosis, they can gather at one particular point, and that is also mediated by actin. And finally, we can also see that there is actomyosin contraction, which can assist the packaging of euchromatin into heterochromatin inside the nucleus of the cell. So that was the role of the nucleus in context of the cytoskeleton, particularly with respect to actin and with respect to lamin. Now we come to another sort of phenomenon where mechanical movements are very important, where cytoskeleton is very important. And of course, the process by itself is crucial for survival, and that is cell division. So what you see here is that you can divide the entire cell cycle into these phases. You can think of them as interphase, S phase, G1 is G2, and of course the M phase and the interphase, which is between two subsequent or two consecutive division cycles. And it is during the process of cell division, or that is during the M phase only, that you can see that there is the formation of two daughter cells. So this entire process is quite, or the different steps or different sub-steps that are part of mitosis have actually been looked at here. So you look at the following phases. You have prophase, prometaphase, metaphase, anaphase, telophase, and separately you have cytokinesis. As part of prophase, we see that chromosomes, they condense, they become visible to someone who is observing it. Then the spindle fibers, which are part of microtubule extensions, they emerge from the centrosomes. And the nuclear envelope will dissolve, it will break down, nucleolus completely disappears. All of this happens during the prophase, and you can see that via the immunofluorescence image as well. Then comes the next phase, which is the pro-metaphase. So in this phase, chromosomes will still continue to condense. Kinetochores would appear at the centromeres. Mitotic spindle microtubules will attach to the kinetochores, and centrosomes will now migrate towards the opposite pole, or they will start migration towards these opposite poles. Then comes the step of metaphase, where mitotic spindle is fully developed. Centrosomes are at opposite poles of the cell, and chromosomes have lined up at the metaphase plate, and each sister chromatid is attached to a spindle fiber which originates from the opposite poles. So if you're already familiar with metaphase, you know that there is formation of a metaphase plate where all of these chromosomes are aligned, and you have spindle fibers which are attached to it from both ends of the dividing cell, or if we want to get into more specifics, then from centromeres present at the opposite poles of the cell, and only after each of the microtubules or each of the spindle fibers have attached to the metaphase aligned chromosomes, only then can the cell proceed towards separation of these sister chromatids during the next step, which is anaphase. So to mediate that, come in the cohesin proteins, and cohesins, they bind the sister chromatids together, which are breaking down, and sister chromatids, which are now called as chromosomes, they get pulled towards the opposite poles. Now, non-kinetochore spindle fibers, they lengthen and they eventually end up elongating the cell. During telophase, chromosomes, they arrive at the opposite poles, and they begin to decondense, and the nuclear envelope material, which will take up the surroundings of each of these chromosomes, mitotic spindle will break down, and then finally a cleavage furrow will be formed through which the two daughter cells would be separated. And for plant cells, a cell plate is present, which is of course not the case for animal cells. In case of plant cells, in plant cells, there is a cell plate instead that separates the two daughter cells. And that is a major distinction between cytokinesis in Animalia and in Plantae. Here you see some representative images of an interphase and a prometaphase nucleus to draw a comparison between these two. So one obvious difference is that the nuclear material or the genetic material has condensed to form chromosomes. Just because it is the pre or the pro-metaphase stage, therefore it has not arranged into a sort of metaphase plate, which we express, which means it is not aligned at the center of the cell, but instead it is dispersed in the form of chromosomes. You can see that it is a much more compact structure, and in fact, what you can also see is that the nuclear envelope is now next to nothing, which is important because you want these chromosomes to be able to not just be restrained within the cell and to be able to arrange themselves at the center of the cell, forming the metaphase plate. So I think there's a definition of it as well. So it says that a series of processes must therefore take place that enables the cell to package DNA within the confines of the nucleus while remaining its ability to transcribe and duplicate the entire DNA sequence, and also maintain its integrity. How is this achieved? So an elaborate process of DNA condensation occurs that sees the DNA packaged into 46 chromosomes, rather 23 pairs in humans. The number of chromosomes will also vary from species to species. And during the process of division, you will see that they get segregated from the 2n state into n stages, and those form the gametes. Here we see membrane and organelle dynamics during cell division, how it changes. So you can see the progression from interphase, prophase, prometaphase, metaphase. You can see how the nucleus itself has completely vanished, how each of these organelles are reorienting themselves based on the type of function. You can see how the centrosomes have now taken up the responsibility of segregating the cell into two daughter cells, and therefore they assemble at opposite poles. And you can see how the microtubules are associated, or how they're attached at the center, and how they divide into the two daughter cells by the process of cytokinesis, and each of them is then capable of further development. Yeah. And here you see a similar sort of arrangement of the same, just simplified into these 5 phases, which are prophase, prometaphase, metaphase, anaphase, and telophase. So we'll move on to the next lecture.
Can I ask some doubt, madam?
Go ahead.
Confusion between chromatid and chromatin. How to differentiate that one? And what is there in nucleolus?
Okay, I think we had this discussion in one of the earlier classes about chromatin, chromatid, and chromosome. Most probably you or someone else, I'm not sure, had asked about it as well. But to give a brief answer, basically the chromatin by itself is all of the genetic material inside the cell. The chromatin is unorganized genetic material inside the cell, right? During the prophase, what you see inside the cell is just haphazard, like they're usually drawn like noodles sort of a format. They are unorganized and um, just genetic material condensed into one region. So that is the chromatid. When you ask about chromatids are organized structures which are part of a chromosome. So let me just switch on my video.
Now,
I
what confusion is, I got confusion. You said now chromatids are called as chromosome in
No,
like
I did not say that. I did not say that. Just give me a second. So what we said was that there are two chromatids. They are forming one chromosome. If you look at that particular diagram, there it is mentioned differently. I'll come to that distinction between them as well. Uh, I don't have anything else to explain it, but basically you have two sister chromatids. This is one chromatid, this is another chromatid. These are sister chromatids. They are carrying exactly the same genetic information, but in copies. They're carrying alleles for each of these genes on themselves. These two chromatids are forming what you see as a chromosome, what you see as an X, which is represented normally as an X. That is one chromosome. There are a pair of such chromosomes which are present, right, within an individual. These chromosomes, when they're undergoing the process of these prophase, metaphase, anaphase, and so on and so forth, when they're undergoing this separation during metaphase, after they have aligned, these, you saw how each of these X's were aligned, they were aligned at the center of the cell. In the next step, what will happen is, sorry, in the next step, what will happen is they will get separated, right? So this separation is occurring such that each of these two chromatids are getting separated at two poles. This is forming one cell, this is forming another cell. This is the process of cell division, the way it is occurring. What is happening as a consequence of this is that what you were previously calling a chromosome is composed of sister chromatids. These chromatids are getting separated. Once they have separated, as per nomenclature, it is not changing what is happening inside the cell, but as per nomenclature, you will find in a large number of books that each of these individually separated chromatids are now also called as chromosomes. I think that was mentioned there in the diagram there as well. That I think I read it out also. Yeah. Here, during anaphase, if you look here carefully, what you saw previously in metaphase in the form of X's, these are individually chromosomes. Read the terminology here. It is said that chromosomes are lined up at the metaphase plate. Now what is happening? Sister chromatids are pulled towards opposite poles. This is what I was indicating, that they are getting pulled towards opposite poles. They are individually sister chromatids that were initially part of the same chromosome. They are getting separated at the stage of anaphase. In terms of nomenclature, in a large number of cases, now these sister chromatids individually are now in a large number of cases called as chromosomes. I hope this clears it up a little bit. Please tell me if there's more confusion.
Madam, simply tell me. Suppose I am having chromosome number 11, so I have two pairs of chromosome number 11. Okay. Now I have four sister chromatids or two sister chromatids.
You have two pairs of chromosome 11, which means you have four sister chromatids.
One pair of sister chromosome. 111, 211 will make one pair. Means I will have one.
Yes, just take this as example. Say this first chromosome that you see here. This is chromosome 11, as per your example.
Yes, and then chromosome 11, then it have one allele, another chromosome 11, so it makes pair of chromosome.
Yes. Say these two chromosomes are chromosome 11, chromosome 11, two copies of chromosome 11. This
11.
1 and 2, right? So
It,
there are
it
four
have
sister
two sister
chromatids.
chromatids. Okay. Now, how these sister chromatids will be called chromosome if they are single, unless they become double?
They're not ideally called so. That is what I'm saying. This is just part of nomenclature. Here you are seeing they are now called as chromosomes. But ideally, no. Chromosome is something which has two sister chromatids. This is the correct terminology. Here they have specifically mentioned it, which is why I am clarifying it to you. That you will find in a large number of books, there is a lot of ambiguity by the nomenclature that is given. Ki same thing is named differently across different books, but conceptually, for your own understanding, you should always keep in mind a chromosome is made up of a pair of sister chromatids. This is very important because, you know, if you have read about recombination, or if you go into the concept of recombination, there is something called as homologous recombination. Homologous recombination, it's a more advanced concept. I'm just bringing in why this terminology and this correct naming is important. That is because homologous recombination occurs between non-sister pair of chromatids. Non-sister chromatids ke beech mein there is the recombination occurs. So if you have chromosome 11, as per your example, one copy of chromosome 11, another copy of chromosome 11, then this and this chromatid are capable, the one chromatid from here and one chromatid of from here are capable of exchanging information among themselves, not between these two, but among these two. So this distinction is important. You can, if you want, I can look up some sites or some blogs, etc., or some papers which explain this in better detail, because some books do have like a little bit confusing nomenclature. But you can go to NCI, PubMed, etc. Preferably go to NIH, NCI sites. They have clear definitions of each of these terminologies. Maybe that would be helpful. And always you can go through Google for images, etc. So chromatids, two sister chromatids make up one chromosome. You have two copies of such chromosomes. I hope that clarifies the whole situation.
Okay, madam, thank you. What about nucleolus? What is there in the nucleolus?
What is there? I will have to look it up. I am not sure. Basically, if you just look at the nucleus, like the simpler arrangement of it, I cannot recall it right now, but the simpler arrangement of it is that if you see a circle, which is the nucleus, there is a smaller subsection of it within the nucleolus, and the remaining area is called the nucleoplasm. One thing that I can think of right now is that within the nucleolus, some specific tRNA processing, I think it takes place, if I am not wrong. Um, yeah, I think tRNA or RNA processing. I'll look it up once again, but the nucleolus has this one particular job that it processes. It's a process similar to that of splicing, where from one tRNA or I think it's rRNA probably. I'm getting it wrong. I'll have to look it up. Most probably it is rRNA, I guess, within the nucleus. From one continuous rRNA, there is processing into three different rRNAs. I think that occurs within the nucleolus. Apart from that, its function, I will have to look up. I cannot recall it right now.
Because in nucleoplasm, you had told me heterochromatin, euchromatin, are
Inside
ribosome.
it, they are all parts of the nucleus. I do not say they are inside the nucleolus.
They are whatever, not inside the nucleus. They are inside the nucleoplasm. So what is there means? What I thought was nucleolus is a condensed part of some means more condensed chromosomes. They are in the nucleolus. That's what I was thinking.
Okay, that could be the case. I will have to look it up. I'm not,
But
I cannot recall it right now.
and euchromatin complex, euchromatin concept has confused me. Suppose heterochromatin is a compact mass of chromosome and there's euchromatin is less condensed. So what should be there in the nucleus? That's why I asked. Another question, madam, what is the nucleosome?
But that is not possible, right? That only the condensed part of chromosome will be in one place. The non-condensed, it's all part of the same chromatin at the end of the day, right? It is only some regions are getting wrapped, some are unwrapped, again some are wrapped, some are unwrapped. It is that beads on string structure is there. So it is all part of the chromatin or the genetic material of the cell at the end of the day. What is exactly within the nucleolus, I will have to look up. I don't think it can be in that way that more condensed genetic material is in the nucleolus. I don't think that makes sense, but still I will not speak unless I have read up on it. So I'll need to look it up. If necessary, I can answer this question on the discussion forum for clarity. Did you have any more doubts? Okay. Not, we'll come to the next part of it.
Only first picture, madam. What is nucleosome?
Sorry, I couldn't hear you once again.
The first slide you had showed, in that you had said some nucleosome. Is that?
Nucleosome we have discussed, right? Uh, okay, so nucleosome, so that beads and string structure that we talked about, the beads part of it is actually what is the nucleosome. The nucleosome has two things. It has proteins and it has DNA. So what are these proteins? These are histone proteins. Histones are like there is a subclassification there. There is histone H1, H2, H3, H2, H3, sorry, I'm getting the nomenclature wrong. H2, H3A and H3B, I think. I'll again have to look it up. It was a while back. So basically there is an octamer of histones, which is 8 histone proteins that form sort of a bundle. I will again use that same example I had the last time. Consider this to be an octamer of histones, which is basically the mouse, and around it DNA is wrapped. Okay? So this is generating what you understand as a nucleosome. This is your heterochromatid. The remaining part of the DNA that is staying behind, that is the euchromatid. This is the simplest way of understanding it. So this is nucleosome. This is the remaining DNA. Again, after some distance, there is another nucleosome. Again, after some distance, there's another nucleosome. That is how it is arranged. So this is why it looks like there is a string, and on that string there are tiny, tiny little beads.
Madam, you
Does that
called
explain?
but there are 5 histones only.
What?
Histone proteins, there are, I think, only 5. 1, 2, 3A, 3B, and
They
4.
form an octamer, which means there are repeats. There is H2. Histone H1 is present at this place. Like, okay, the point where DNA stops wrapping, histone H1 is there as sort of a clip, basically. It is clipping that this is where the wrapping will stop. Within the octamer, there is histone H2, H3A, H4, I think something like that. That arrangement is there. H4A, H4B, or H3, H3B is there. So there are 4 proteins that are present in duplicates, and that forms the octamer. I can open it up just to clear it. Okay, it's clear, then fine. Okay, so yeah, we discussed this already, but yeah, this I think also clarifies what you were talking about. That chromosomes, they consist of 2 sister chromatids. They are linked at the centromere by cohesin proteins. Cohesins, if you see the most accurate representation of cohesins is in the form of a belt. As the name indicates, they are cohesively holding, which means they're gluing something together. The 2 sister chromatids, they are glued together or are cinched together by the cohesin proteins, and that is occurring at the center of the chromosome, which you can see in this electron micrograph as well, that they are forming this sort of cinched structure, and that portion is what you understand as the centromere of the chromosome. So this is during the metaphase. During the early prophase, what you see is the more disorganized sort of structure, more open, more dissociated sort of structure that we were previously talking about. And here also you can see that at the centromere, you can see that there is attachment of kinetochore microtubules, the plus end of it. And the architecture is also represented schematically here, where you can see that you have the 2 centrioles at the 2 ends, 2 centrioles, two centrosomes at the 2 ends of the cell, which each of these, they contain 2 centrioles and the pericentriolar material, like we have already talked about. And from there, this also, I think we have talked about the arrangement of microtubules during the process of cell division, and how you can see that microtubules, some of them are projecting outside. Those are the astral microtubules. Some of them are, overlapping with the microtubules from the other centrioles. Those are the overlapping interpolar microtubules, and some of them are attaching to the chromosomes, and to the different sister chromatids of each of these chromosomes, and they're helping in pulling the two apart. So interpolar microtubules, they extend from the spindle poles and they interdigitate. Interdigitate basically means, consider these as 2 digits. These 2 digits are overlapping with each other, and they are sort of interspersed along or with respect to each other. And what happens is that it stabilizes. The 2 interacting microtubules become stabilized. They are not undergoing dynamic instability as they normally do. On the other hand, the dynamic astral microtubules, they extend from the spindle poles radially towards the other side of the cell, or towards the plasma membrane. And this we have already looked at, that during prophase, which is the first step of mitosis, the microtubules will rearrange and they'll begin to polymerize to form a bipolar spindle as DNA is condensing into chromosomes. So during prophase, microtubules are forming a spindle sort of structure during which DNA, sorry, just yeah. During the during prophase, the first step of mitosis, the microtubules they rearrange and they're forming the spindle. Also, DNA is condensing into chromosomes. And the cells they exit division during the process of telophase. The microtubules will again condense to form an intercellular bridge or a mid body, which you can see here. Because during telophase, the objective is to carry out the cinching or the snipping of the bigger cell into the two smaller cells. And this is where the separation of the daughter cells takes place. So microtubules have a major role to play till the end of cell division. And here you can see the microtubule, the centrioles and how they are actually undergoing the process of duplication. You can see there is a mother centriole and there is a daughter centriole which is there. They have proximal and distal ends. They have some interconnecting fibers. This is again a lot of detail, but just for your information for the time being, you can look at it. And how the centrosome also undergoes cycling during the process of cell division has been shown here. So first step is of course centriole disengagement, where the titling between mother-daughter centrioles becomes severed. They are connected simply by some loose fibrous structures. Second, the centrosome undergoes duplication and new centrioles form at the proximal ends during S phase when cell is duplicating its DNA. Then there is engagement of the centriole where the newly formed ones will reach their full length and the other daughter centriole becomes the mother now. And the link between the two is severed. Then comes maturation of the centrioles, where the two pairs, you have two mothers and two daughters, they are now forming some, or they are now collecting some additional pericentriolar material, or PCM. And then during the prophase, the centrosome, they generate spindle fibers between them and they move away from each other. So from the original centriole to, or from the original centrosome, two centrosomes have now been formed and they are migrated towards opposite poles of the cell, and they are resulting in the formation of a bipolar spindle that is beautifully represented here as the metaphase plate. You can see the chromosomes aligned at the center, indicated in blue, and at the two ends, you can see the um, two centrosomes that are present, indicating the bipolar spindle. And finally, the cell will divide, and each daughter cell will receive one centrosome with a pair of centrioles. And when that cell undergoes division, again, that pair of centrioles will undergo duplication and they'll form two centrosomes for the cell. The rearrangement of the actin.
Centrosome and centrioles, one and the same thing or different?
No, they're different. It's very, this entity, but I had shown it in one of my previous slides also. Let me show you in this way. Can you hear me? Hello?
Yes, madam, I can hear you, but
Yeah, I
now
thought I got disconnected. Yeah. So I was saying basically, that you have a centrosome. Think of it as this entity. What it is made up of is two centrioles, which are oriented at an angle with respect to each other, and they're surrounded by some sort of cloudy material, which of course is made up of certain things. You can think of it just as a cloudy material for now, that is known as the pericentriolar material. So you have two centrioles and the pericentriolar material altogether forming the centrosome. Each cell initially receives one centrosome, and at the time of division, this centrosome is duplicating and forming two centrosomes. Those two centrosomes are moving towards opposite parts of the cell. From them, the spindle fibers are emerging, and those spindle fibers are pulling apart the chromosomes at the time of anaphase. Again, each daughter cell ends up with one centrosome that will again undergo duplication, and this process will continue over each cell division. Is that Did you get it?
What I got is centrioles with pericentral material is centrosome, and from centrosome, when they are separated, they become centrioles.
No, they are still a part of the centrosome only. Centriole, what you see, these little like pasta-like things that you see, they are individually centrioles. That is always associated with the pericentriolar material and it forms the centrosome bites. It's not a membrane-bound structure. It stays like this only. That is the centrosome. That is within that centrosome, those individuals, these two, think of these two, those pasta-like materials, like I said, they are each duplicating. They are forming from two, they are forming four. Those four are gathering more pericentriolar material and becoming a large entity. From there, they are snipping and they're becoming two each. They are forming one centrosome, another centrosome, migrating towards opposite part of the cell. Each of those centrosomes has centrioles and the PCM, both. You can go through this diagram in more detail. It's very beautifully explained. You can go through this at your own time in more detail.
Madam, specific asking question is that sperms along with the DNA material carry only centrioles, not centrosome, to the ova. That's why I was confused.
So, okay. So they can gather pericentriolar material from the ovum, maybe. That is possible.
Because
We are not
sperms
talking about fertilization here. We are talking about cell division.
we are talking about cell division, okay, madam. But this cell division, when meiosis is happening, that time centriole is centrosome or centriole plus pericentral material. That's why I am.
Centriole plus pericentriolar material is the centrosome. That is what, that is, I'm in agreement with you. That is forming the centrosome. I'm not understanding the confusion here.
No, thank you, madam. I got it, but means in because what question is usually asked is what is carried along with nuclear material when sperm fertilizes the ova. The question most of the time it is asked. The answer to that is in option. If centrosome and centrioles both are given, we have to choose centrioles, not centrosome.
Okay, yeah, that is a separate direction. That is completely separate. So in that case, it is not bringing PCM along with itself. The male gamete is not bringing PCM with itself. It is just bringing the centriole. So the answer to that is okay. What we are talking about here is what is the centrosome. Then we are talking about the fact that it has both centriole and the pericentriolar material.
Okay, thanks, madam. Please continue.
Yeah. So I think paper here, the coming back to the cell cycle stages again. So rearrangement of the actin cytoskeleton during division is coordinated with the microtubules. During mitotic entry, there is rounding of the cell, which is caused by changes to the actin cortex. And during the process of anaphase, a cleavage furrow is formed, like we've already discussed. This is the cleavage furrow, which is indicating that the cell now needs to undergo some sort of split. And that is mediated by actin, myosin 2, and several other proteins. In fact, in some cases, actin bundle is also indicated towards more later stages, towards the telophase stage. And during it starts to form at the equator of the cell, and during this process, the ring is slowly ingressing during telophase, and it provides the mechanical force that is required in order to split it into two, and then that mid body is also severed, and cytokinesis will then be completed. Here we are seeing that dividing cells. I think this was not elaborated, was it? Yeah, this was elaborated. Yeah, so dividing cells, they regulate the lipid composition and the lipid localization with them. For example, sphingolipid biosynthesis perturbation results in cytokinesis failure. The roles of phosphinoinositides in cell division are beginning. Okay, this is not that relevant to us. Basically, what the understanding is that lipids themselves also have a role to play in cell division. It is not just, you can't just divide the nuclear material of a cell and be done with it. All of the organelles have to double. The lipid material of the cell, that also has to double, and therefore there's a separate lipid biology that is also studied with respect to cell division. Here you can see it in more detail. So during mitosis, there are mitotic kinases that phosphorylate a large number of proteins, and that enables disengagement of the nuclear envelope from the lamina and chromatin, releasing the cytoskeletal contacts, and that dissolves the nuclear lamina, which is why you are able to, like, it is one of the markers of prophase that the nuclear lamina is completely dissolved. The phosphorylation of the nuclear pore complex nucleoporin subunits causes this dissolution and the distribution of nucleoporins into the ER and the cytoplasm. During this process, farnesylated, a type of post-translational modification of lamin B, remains in the mitotic ER, whereas the non-farnesylated was farnesylated, version is released into the cytosol. Separate ER and nuclear envelope identities are lost during mitosis, and they only reform upon exit from cell division when the activity of some phosphatases reverts the mitotic state and allows the regeneration of the ER and the nuclear envelope. So this is what happens to the lipids over this course of time. Again, it is a lot of detail, but for now you can just understand it as it is, that there is entire dissolution of the nuclear and the ER identity, nuclear membrane and some of the associated ER. And after mitosis, again, there is regeneration of these two envelopes, and no individual identity is maintained at that point of time. During division, the kinases that were activated during the M phase, they phosphorylate Golgi localized targets, which are shown here. This disrupts the homodimerization of the grasps and dissociates Golgins. These lead to unlinking and disassembly of Golgi stacks and contribute to the suppression of incident vesicular traffic. The membrane fission protein, which is CTPP1 bars, probably one of the options from the previous week's assignment as well. It separates the Golgi into isolated mini stacks, and the release of GTPases like ARF1 suppresses Golgi structure, identity, and function. There is a block in vesicle fusion, which leads to the formation of small tubular vesicular clusters referred to as Golgi haze, allowing Golgi membranes to be inherited through either stochastic mechanisms or by associating with the mitotic spending. So the long story cutting it short is basically what here we are looking at what is happening to all of the other organelles during the process of cell division. We have extensively studied about the nucleus, what happens to everything else. So among them, we are seeing that the nuclear envelope and the ER identity is lost. It is completely dissociated. And after the process of mitosis has been completed, its identity would again be, or these organelles would again be regenerated and their identity would be restored. Next comes the Golgi. For the Golgi, we are seeing that there is disassembly of the Golgi stacks, and there is formation of something known as a Golgi haze, and that is brought about by one of the major contributors to this process is CTBP1 bars, which is a membrane fission protein. By fission, you obviously understand it is breakdown. It is breaking down the membrane, and it is forming these mini stacks, and it is releasing certain enzymes which are suppressing the Golgi identity, structure, and function. All of this would be reassembled after the process of mitosis has actually completed. And here we see it is again further organization of the cell that is occurring during mitotic entry. The next organelle that was looked at was the mitochondria. For mitochondria, there is a little bit of a different process is followed. At the time of interphase, mitochondria are more elongated-like structures. When the cell enters mitosis, there is a fission protein which comes in. You can see just these images for your understanding. You're seeing that during interphase, there are longer mitochondria. However, certain proteins like CDK1, aura, etc., all of these, they snip them or they break down the mitochondria into tiny bits, and you can see that they get arranged during the process of mitosis at the cellular periphery or at the exterior end of the cell. And these fragmented mitochondria are divided throughout the cell, and when the cell exits mitosis, mitochondria begin to refuse, and that is driven by certain types of fusion proteins like mitofusin 1, mitofusin 2, MFN1, MFN2, and that eventually leads to their reassembly. And during the process of cytokinesis, the mitochondria are mostly fused and they are more enriched at the cleavage furrow, possibly because the process of cytokinesis is quite energetically expensive, and that would could potentially allow a larger supply of ATP. This is just something I'm guessing. I'm not sure of it. And this was, I think, a video that was shown about how actin cables and comet tails, they organize mitochondrial networks in mitosis, where they had monitored that within the cell over a period of, I think, 10 or 15 minutes, they had observed that actin actually shows a sort of swirling motion inside the cell, and there is an equal movement of mitochondria inside the cell as well. Possibly that is how you can see that they are getting reoriented around the cellular periphery during the process of mitosis as well over the period of division. So that is what was seen. So to sum it up, you can see that there are different changes that are occurring at the level of not just the nucleus, but the lipid membrane, nuclear membrane, ER, mitochondria, Golgi, and of course the cytoskeleton as well, all of which is undergoing rearrangement at these stages to produce the daughter cells, and each of these have been separately studied. Anything that interests you, you can certainly go back and look at it. So I think with this, we are done with the material that was covered as part of this week. So I'll just wrap up whatever has been discussed till now. So as part of this course, you first learned the central dogma, how it was discovered. You learned each of the different steps of the central dogma. We looked at transcription, translation, replication, each of these processes in detail. We looked at the molecular machinery which operates in that case. We understood a little bit about genetics, Mendelian genetics, non-Mendelian genetics, epigenetics. Then we moved on towards the cell biology aspect of it, and we looked at the cell membrane. We looked at the process of division. We entered into the cytoskeleton, each of the elements, how they function, how their misfunction or dysfunction can trigger different types of disorders. And finally, we ended with focus on the endomembrane system and how each of these have different functions apart from the designated ones, and how relevant they are during the process of cell division as well. So all of this has comprehensively covered at least the initial topics of cell biology and molecular biology. So I think from my end, that would be the completion for now. If you have any doubts, please go ahead. We can wrap it up in the last 5 minutes and then we can end the class.
Madam, two doubts. One, number of mitochondria is fixed into the cell. Why each mitochondria has to be divided into two? Since I don't understand. Ideally,
No, those mitochondria during interphase are more elongated structures, na? Mitochondria is first becoming bigger and then dividing it.
and second, what is that plus and minus in the microtubule? Does it has any role while cell division? Because you said microtubules play very important role during mitotic division.
If you go through the cytoskeleton lectures, we have discussed a lot about it actually. The plus and minus ends are because of the arrangement of the microtubule subunits. So it is made up of
That,
tubulin. So the way in which
that I got
what
ki plus end is towards plus end is towards the plasma membrane and minus end is towards the nucleus. But during cell division, what happens to that plus and minus? That's what I am asking.
nothing happens to it. It is basically it is the orientation of the microtubule that is present during the process of cell division. And based on the orientation of the microtubule, there are certain proteins which are microtubule motor proteins that will move in one direction or another based on how the orientation is made. If you go through it, you will see kinesins and dyneins. They are plus and minus and directed proteins which are present during the process of cell division. So much is occurring, right? You are just not organizing the chromatin. You are shifting a lot of cargo inside the cell. All of that is taken
Madam,
care of by these motor proteins, and therefore the arrangement or the orientation is made in that manner. And also another major thing that occurs is during the process of cell division, there has to be one of the major steps which is actually helping anaphase to take place is that there is disassembly of the microtubule from one end, right? For that disassembly, the orientation is very important because it has to depolymerize from one end in order to shorten. That microtubule has to shorten to pull those two chromatids, sister chromatids apart. So there also the orientation is very important.
because kinines always will move from plus to minus state, whereas dyneins will always move from minus to plus side.
They are a family of proteins. Most of them do follow this, but there might be exceptions to it as well. The kinesin family is there, dyneins family are there. So there might be variations within it, but what you said is majorly how it is classified, yes.
Thank you, madam.
So if there are no more doubts, then thank you so much for joining and all the best for your exam. And thank you for attending all of the courses and all the best. Thank you.
Thank you, madam. Thank you.